This is a brief tour of a web application for exploring data on genetic variation in 3 crosses of the malaria parasite Plasmodium falciparum. To access the application, first visit the P. falciparum genetic crosses homepage. From there, we can navigate to the latest data release, and from there we can access the web application. From the intro page, you can access various tools for exploring the data. In the sample metadata, you can access information about the parasite samples that have been sequenced. For example, for the cross between clones 3D7 and HB3, there were 21 samples sequenced in total. For each sample, you can see the sample ID, the clone ID, the mean depth of sequencing, and there is a link through to the European Nucleotide Archive where you can access the raw sequence data. In the variants table, you can access information about the SNPs and small indels that were found within each of the crosses. 2 separate methods were used for variant discovery. The GATK method is based on alignment of sequence reads to the reference genome. The Cortex method uses de novo assembly to discover variation. For each method, there is a table of variants discovered. You can see the variant position, the gene in which the variant falls, if it does fall within a gene, the reference and alternate alleles, whether the variant passed any filters, the predicted effect of the mutation, and various other fields. By default, only variants passing all quality filters are shown. However, you can disable these filters if you like to see the raw data. Let's reapply the filters. So in the genotype browser, you can view individual genotypes for each of the parasite clones in a cross. So here we are viewing genotypes for the 3D7 HB3 cross, and here every row corresponds to a sample and every column corresponds to a variant, which is either a SNP or an indel. Here I've zoomed out to the whole of chromosome 1, and you can zoom in or out to particular regions of the data and pan up or down the chromosome. Here I've zoomed into an individual gene. Each cell in this view is a genotype call, and by default the genotype calls are coloured according to the allele of the parent. So here blue alleles correspond to 3D7 and red alleles correspond to HB3. And this allows you to visualize the pattern of meiotic recombination in each chromosome. For example, if we view a larger chromosome, such as chromosome 14, we can see that more recombination events have happened. In the genome browser, there are various tracks which provide information about genes, variants, sequence read alignments, and other data. For example, let's look at chromosome 4, and let us access coverage and mapping quality data. So here on chromosome 4, you can see that coverage and mapping quality are very different in the towards the ends of the chromosomes, in the subtelomeric regions of the chromosomes, but also in 2 regions within the chromosome. And these correspond to regions containing the hypervariable VAR genes. So this view allows you to explore some metrics which give you a sense of how accessible the genome is in different regions. If you would like to browse the sequence alignments directly, you can use the Look-Seek browser. So here you can select individual samples and a view of all of the sequence reads will be rendered, and you can use this to zoom in on particular genome regions and, for example, view read pileups. You can also use this to view coverage data. For example, let's take a look at DD2 on chromosome 5. And it's coming in a minute. Here we go. On chromosome 5, you can see that DD2 has a copy number amplification in this region of the chromosome, which spans the multi-drug resistance gene MDR1. Finally, all of the data which are available through this web application can also be downloaded directly from an FTP site. For more information about this web application, you can visit the data release webpage, and there's also a preprint available on bioRxiv which describes the data, the methods we use to construct it, and various other features that we've discovered. Thanks for watching.